This video demonstrates a procedure for separating mixed viral assemblages based on their nucleic acid composition. This enables the study of the taxonomic diversity and functional potential of complex viral assemblages. Here, hydroxyapatite, a crystalline form of calcium phosphate, is employed in the separation of nucleic acids. First, a hydroxyapatite column is packed and equilibrated. Purified viral nucleic acids are then applied to the column. The charge interaction between the positively charged calcium ions of the hydroxyapatite and the negatively charged phosphate backbone of the nucleic acid subtypes binds the nucleic acids to the column. Next, single-stranded DNA, RNA, and double-stranded DNA are sequentially eluted with different concentrations of phosphate buffer. The resulting nucleic acids are then desalted and concentrated in preparation for visualization, quantification, and identification by DNA sequencing. Agarose gel electrophoresis confirms that the nucleic acid species can be separated with less than 9% carryover. Thus, this method enables the evaluation of the diversity and functional capacity of viral communities.
Hello, my name is Doug Fadrosh, and I'm a research associate in the lab of Dr. Shannon Williamson at the J. Craig Venter Institute. Today, I will be showing you a procedure to separate single-stranded DNA, double-stranded DNA, and RNA from an environmental viral assemblage. This method has advantages over existing methods, such as the treatment with the nucleic.